Chapter 4, Section 2, Growth Media and Laboratory Culture. When you're in the lab, a microbiology lab, either here at school, a hospital, a research center, a pharmaceutical company, whatever, you're going to see that you need to be able to culture microbes, bacteria, fungi, yeast, whatever. To culture them means you need to grow them under set conditions. Well, one of those set conditions is that you're going to have to supply all the nutrients, like we talked about in Section 1. The thing is, you can't just mix everything together in water, because the powders and things that you're going to be mixing, well, they've been exposed to dust. There's going to be bacterial endospores, fungal spores. The water you're going to use, well, going to have bacterial spores, endospores, all sorts of stuff. So whatever you make, you're then going to have to sterilize. And to sterilize something means not only do you kill off any living cell, the vegetative cells, you also have to irreversibly damage all spores so they cannot germinate and release vegetative cells. We generally do this with what's referred to as an autoclave. Autoclave, think of these as industrial-sized pressure cookers, because that's what they do. They put everything, your culture media, your glassware, your instruments, whatever, put them under extreme pressure, extreme moist heat. Things get damaged, things die. And as you're in the lab, we'll see that we have different media. Remember media, I told you back in Chapter 1, when I say media, this is discussing the different types of broths and augers that are available for culturing bacteria. Well, we have different media based on composition. Do we really need to know everything that is there? How much potassium phosphate? How much, ammonia sulfate? How much magnesium sulfate? How much glucose? What are the trace elements, and at what concentrations are each one of these? Or we put some sugar in, 15 grams. Yeast extract, which means we took a bunch of yeast, dried them down, ground them up. So whatever amino acids, whatever lipids, whatever nucleotides, whatever sugars, whatever was in the yeast, it's now there. Same with peptone. Peptone is the enzyme peptidase digest of, some meat. There you go. There's just a bunch of amino acids. Which ones? Don't know. Could be all of them, could only be some of them. This is what is defined as, quote unquote, defined culture medium. We know exactly everything that is there. Every component that is added to that water, we know. What metals, what trace elements, how much of each, what sugars, how much, we can calculate. The concentrations of glucose there. We can calculate concentration of calcium, ammonia, potassium, all of that. Over here, with what was referred to as a complex, we know 15 grams of glucose, 5 grams of yeast extract. Okay, of that yeast extract, how much of it is amino acids, how much of it is proteins or polypeptides, how much of it is phospholipids, how much of it is this, how much, don't know. It's just too complex. Hence the name, complex. So the defined, we know the exact composition down to which elements, and we can calculate percentage. We can calculate, the millimolar, the micromolar concentrations. The complex, it's a bunch of digest. We use enzymes to digest down, different tissues, releasing the amino acids, the nucleic acids, so we can get at the sugars, releasing the lipids, so we can break them down 2 by 2 to add, to feed into and do all sorts of stuff. Couldn't tell you how much and of what, because it's just complex. We take this a step further, and we can go back with, say, the defined media, or some of these complex medias. And we can add certain things to them, tweak them just a bit. We tweak them so that only certain things, certain bacterial species, certain bacterial groups can grow, and all others will be inhibited. Not that they can't grow, but it'll be really hard for them to. We call this selective. It selects for one type, inhibits all else. We add bile salts to certain augurs, and we'll talk about those augurs in lab. Bile salts is selective for enteric bacteria, bacteria that would be found in the gastrointestinal tract of an animal. All other organisms that have not spent hundreds of millions of years evolving to grow in the enteric system, the gastrointestinal system, will be inhibited by said bile salts. Or we can add in certain indicators, dyes. We call these differential media. What is it? EMB. E. coli turns one color, all other enterics turn a different color. It allows you to tell, is this bacteria I'm working with, I am trying to identify, is it E. coli? Is it something else? Is it a different gram-negative enteric bacteria? We find that different organisms are going to have different requirements. So if we try to go too defined, yeah, we can go too defined, where here's a media that is everything Staph aureus needs at exact concentrations. Well, good, it's great for Staph aureus, but what about strep? What about micrococcus? What about something else? What about bacillus? Other gram-positives? May not work. So we can go as defined. Certain ones you're going to have to go extremely defined. Neisseria gonorrhea is going to require chocolate agar. We'll talk about that, adding in certain things in a few minutes and a few slides from here. That's going to be one, the red blood cells are burst, releasing the heme. And, factor X and a few other things there. Yeah, that is a specific agar for a specific bacterial species. Can other things grow on it? Maybe, maybe not, but So yes, we can go extremely specific, but we try to go as broad as possible. So that way, one type of agar will allow for multiple species to be able to grow on it as we're doing different things. So we don't have to have, if we're working in a lab with 4 different types of bacteria, we don't have to have 20 different types of agars for different times and different conditions and things like that. 1 generic or 2 generic agars will allow for us to cultivate the 4 we're studying, and then maybe any others we want to add in at a later time. You also have to take into account whether you're going to be doing liquid or solid media. Solid media is thickened by agar. Okay, you saw this in lab when you were doing the streak plate. Had the consistency of really firm jellos because this agar grabs and holds the water in place. So there's lots of water there. It's mostly water. It's all held in place by agar. It makes a gel that's, like I said, it's almost like a thick jello. It allows the growth of cells on the surface, known as colonies, which is a good thing, because as we discussed in lab, if I'm going to do any type of, assays, biochemical assays, genetic assays, things like that, I need to have one specific species. I can't run the risk that my tube has multiple different types of species present, because that could throw off my results. Colony always starts with one bacteria. One bacteria that, given enough time, will result in millions of its clones all growing on top of each other. I can't see one bacteria, it's too small, but I can see millions of its clones all clumped together, and we call that a colony. The way the colony grows, its color, its texture, are characteristic for the most part for different species. What color? Staphylococcus aureus takes on a golden hue color, which is the name aureus, golden. Streptococcus pneumonia has a slime layer, so its colonies will be nice and thick and gooey. Sometimes they're going to take on specific colors because it turns out the bacteria excrete certain pigments. And when you go and you look at here, and figure 4.2 in the textbook, you look here at, what is this, panel E. You look across here, and this is a mixed culture, and you can see many different colors there. Whites, different shades of whites, different shades of yellow, red. I mean, nice pinpoint colonies, big colonies, or this, cloud-looking puffy colonies. Each of the colony characteristics, as they're showing here, big, puffy, red, more defined. These are all driven by the cell's metabolisms, which are driven by their genetics. And the genetics are different species to species. We have to keep in mind, one of the things that are going to be paramount in your mind in the lab is everything that you're going to be starting with when you start a lab experiment, when you start a research project, or if you're in industry, you start your work, your project, your day, whatever. Everything is sterile, and you want to maintain sterility. You do so by what is called aseptic technique. You're doing everything you can to minimize the chances of contamination. Contamination from the, whatever's floating around in the air, dust, which on every dust is going to be bacterial spores, fungal spores, contamination from you because, well, you're dirty, you're filthy. As we've looked at in lab, since you don't know what is always going to be there in a tube, you always want to try to start off with a streak plate. A streak plate, which will give you one colony of one bacterial species, millions of clones of that one original that you can start doing things with. This figure's a little outdated from the textbook. We don't use Bunsen burners anymore. The open flame, we're trying to get away from that because that open flame, I honestly don't know why. I think it's somebody sat down and thought, oh my God, that could be a hazard. You flaming something on that little bitty loop, it could aerosolize, you could breathe it in. Oh my gosh, you could get sick. Not that it's ever happened that I know of, not that it's ever been reported that I know of. But hey, somebody, got paranoid, and now we all have to use those micro incinerators. But notice, the loop, wherever you got it from, unless you took it out of a wrapper that, was guaranteed by the manufacturer to be sterile as long as the wrapper is intact, it is dirty, it is filthy, it is contaminated. By heating it up, you're basically carbonizing, charring everything there. You can now dip it into stuff, move stuff around. And As long as you maintain not touching the loop to anything else, not touching the loop to you, you not touching the loop, you not touching down into the tube and whatnot. You are maintaining an aseptic technique. You are maintaining a level of sterility. You look here. Here is the OG way of doing a streak plate. You take the loop, you sterilize it, you dip it into your broth, and then you smear it around. Re-sterilize, 1, 2, 3, boom, sterilize, 1, 2, 3, sterilize, 1, 2, 3, sterilize, 1, 2, 3. Problem is, when you're doing it this way, I mean, this works, as you see here, it works great. Problem is, a lot of times I've noticed, and it happened to me too, students get streak happy, and instead of going just here to there and stopping midpoint, they go all the way over to 1 and then come back. And so they go, high numbers, lower numbers, lower numbers, lower numbers, lowest, and then they accidentally go back over to here to 1, streak 1. And then they pick up from the high numbers again, and then they wonder why, they have so many bacteria that should be. So that's why, I came up with the idea of drawing the quadrants. So that way you go from one quadrant to the next, to the next, to the next. You never cross back over to any ones you've already went through. Bob's your uncle.